So we have these, it's called the light-dependent reactions, but a lot of people just refer to it as the light reactions. Okay, because it's really dealing with the sunlight coming in. Okay, so either or, you're reading in your book and other places, light-dependent reactions and light reactions are referred to the same thing. I'll show you what the other one is and what they call that. So in the light reactions, you have photosystems. And in one unit, you're going to have two different photosystems. So what's a photosystem? They're located in the membrane of thylakoids. Something very similar to what we've once seen with electron transport chain, with molecules, that was 4 different molecules that were embedded into the membrane. Kind of a different system, but it's similar to that. Okay, you're going to have electrons being transported here. The light reactions occur in these photosystems, these units. And these photosystems are made up of, remember, MUO is made up of proteins, and inside there will be chlorophyll. The chlorophyll is the things that are going to absorb that energy, that solar energy. So each photosystem will contain 2 parts. There's something called the reaction center complex, and that's the place where light energy is actually converted to chemical energy. Okay, but the light needs to be funneled into this reaction center. So the chlorophyll is here, that's the one that's going to absorb the light, okay, and it should transfer as an excited electron to an electron acceptor. Alright? So the reaction center is where the actual conversion of light energy going into chemical energy. But we also have, if you think of it like the center part of this photosystem is going to be the reaction center, there's also going to be proteins all around it, surrounding it. They're called the light harvesting complexes, which basically is just going to funnel in this light. All right, so they kind of surround it. Think of it like, let's see if I can do this. If this is so these are the harvesting, the light harvesting complexes. Think of it 3-dimensionally all around this. So there's coming out of here too. But the center part over here is going to be the, as the name implies, is going to be the reaction center. So what's happening is that these harvesting light complexes kind of funnel in the light to make sure it goes into the reaction center. Okay? What it looks like is that. So what you have here is, this is the center part. All right, the part that's going to actually do make the chemical energy from light. But then there's going to be these light harvesting complexes, these green things all around here. And there's chlorophyll in here that are going to be absorbing that light. There's going to be some chlorophyll in here too. What's happening is the light is going to go into these harvesting light complexes, and it's going to funnel in to where the actual chemistry is going to happen. All right, and this is a photosystem. All right, you can see this wonderful phospholipid bilayer. All right, this is where the thylakoid membrane is. The stroma is out here, and inside the thylakoid, we call that the lumen. I think I've used that word before. Lumen is the hollow area of the cavity of a structure. You have a garden hose. The water goes through the lumen, through that cavity, right? All right. So the thylakoid lumen is that cavity inside the thylakoid. Questions about how that is structurally made up. All right, so we have two types of photosystems, photosystem 1 and photosystem 2. Sounds very simple, and they have better names, but we're not going to be calling that here in the class. It'd be easier just to call it photosystem 1 and photosystem 2, which in reality that's what they call them there too. There is a longer word for each, you don't have to worry about it. Each photosystem has a unique electron transport chain. Yeah, that's coming back up again. All right, so it's a series of these electron carrier molecules that happen in between the photosystems. All right, so the pathway of the electron will flow from photosystem 2. Now that's an odd thing. Like, why are we starting with photosystem 2, electron transport chain 2, and then goes to photosystem 1? Because this is how they were discovered. So they actually discovered this photosystem 1 first, and then many years later they said, wait, there's a one that happens before that. So they end up calling that one photosystem 2. Because you can't change it, because all the books have already called this photosystem 1. Does that make sense? And it'll confuse people and say, well, which one did you mean? You mean the old photosystem 1 or the new photosystem 1? Does that make sense? So this is the reason why, is because of the way they were discovered. So it goes from, the light goes into, or the electrons go from photosystem 2, it'll go through an electron transport chain, we call that part 2, and then you have photosystem 1, electron transport chain 1, and then you will get the NADP, right, on NAD. So, and I'll show you a picture of it. So we have this light-dependent reaction, okay? It occurs in the thylakoids, and this is what is happening. It's taking water and light and changing it into chemicals. So you're taking the light and changing it into chemical energy, ATP and NADPH. Okay? And this is where oxygen comes off. This is where the plants actually are going to give off oxygen that we as humans and animals need for our cellular respiration. That's where it's coming from. So oxygen is coming from the light-dependent reaction, or light reaction, if you want to call it that. Carbon dioxide, you all know that carbon dioxide plants need. Well, this isn't the part where carbon dioxide comes in. That's in the second part, the part where we're going to talk about the Calvin cycle. So light energy powers the electron transport chains to make electrons flow through the systems. Okay? Something similar that what we saw with the electron transport chain in cellular respiration. And this in turn will pump the hydrogens, or protons, from the stroma into the thylakoids against the gradient. So very similar to what we saw in cellular respiration. Okay? But in cellular respiration, here's where it's a little bit backwards. If you remember electron transport chain in cellular respiration, it's putting and it's in the mitochondria, you're pushing hydrogens from the matrix, the inner portion of the mitochondria, and you're pushing against the gradient into the intermembrane space. This way, it's going the opposite. You're pushing the hydrogens in the thylakoids. You're pushing the hydrogens from the stroma, which is outside the thylakoids, and you're pushing it into the thylakoid space itself, or the lumen. So it's kind of doing the opposite. So photosystem 2, that's where we're going to be breaking water molecules. This happens in the thylakoids, and what's going to happen is it's going to break water. So it's like 2 molecules of water. You're going to get 4 hydrogen protons, and that's where the oxygen comes off of. So the oxygen is coming off of breaking the water molecules inside the thylakoids. That's where the oxygen comes from that we breathe in. So that's why plants need water, so that we can breathe. Okay? So this creates a very high concentration of hydrogen protons or ions now in the thylakoids, right? NADPH is made, and we also have the wonderful ATP synthase, which is going to convert those, using the power of high concentration of hydrogen ions, and through just simple diffusion, it's going to run through the ATP synthase to make ATP. It's going to convert ADP and inorganic phosphate to make ATP. The hydrogen protons coming in is going to power the ATP synthase, just the same way that you saw with cellular respiration. Okay? And it's going down its gradient, going from a high concentration of hydrogen to a low concentration. All right? And ATP is made, as the name implies. All right, so let's show you what's going on over here. It's similar to what you saw with cellular respiration with the electron transport chain in the mitochondria. Let me orient you. There's a chloroplast, and you have those stacks of thylakoids, those stacks of green pancakes. It's called granule, right? We're just going to take one membrane of a thylakoid, and that's what this is here. It's a phospholipid bilayer. In there, you're going to have two photosystems, photosystem 2 and photosystem 1. We also have two sets of electron transports. One will be here, and one will be here. What's going to happen, let me show you also, this is where the stroma is, so this is outside the thylakoid. This is the interior area of the thylakoid lumen. What's going to happen here is that light comes into this photosystem 2 by way of the harvesting light complexes. They're going to funnel it into the center portion that's going to do the reactions. And when it does, it's inside, it's going to take water and split it. It's going to split it, as the electrons go in here, it's going to split it so that there's going to be more hydrogens inside the lumen. Now that's the opposite of what you saw before in electron transport chains in the mitochondria. Usually you were seeing a lot of hydrogens outside. In this case, it's putting a lot of hydrogens inside. This light is going to take the water, split it. Hydrogens will stay here. The electrons will then go through this system. The electron will go into another system here to push more hydrogens from the outside to the inside. More light goes on here, so we can power the electrons to keep on moving. And it's going to take NADP P and convert it to NADPH. You're going to oxidize it, right? The way we saw it with NAD and NADH. So we're going to take the NADP, oxidize it to NADPH. Meanwhile, all those hydrogens are being poured in here. It's going against the gradient. Normally, there's a little bit of hydrogens, there's a little bit of hydrogens here and a lot here. But this whole system is active transport. It is putting more hydrogens inside the thylakoids, creating this huge pressure of hydrogen ions inside that want to burst. They want to get out. There's too many in here. You have 2 people, you have, let's say, 10 people outside the room. You have 100 people in here. You got to squeeze those other 10 people in here. This room is going to burst, right? Once we open up the doors in the back, you're all going to escape out there. That's what's happening here. We're creating this huge concentration of hydrogen ions. The only place it could do to open up the doors, the only place they can go out is here. ATP synthase. There's a little opening here, and these go right through there. And it's going to go from a high concentration to a low concentration. That's simple diffusion. As it goes through here, it makes this ATP synthase kind of turn. So we're using the power of hydrogens to go from a high concentration to a low to power this ATP synthase to turn. And as this, every time it clicks to turn, it's going to take ADP and an inorganic phosphate and make an ATP out of it. This is called chemiosmosis. All right? So what we have here is a few things going on. We're having an electron transport chain, which is here. There's a couple of them over here. But this is not part of the electron transport chain as much as in mitochondria it wasn't either. This is called chemiosmosis. It's using the power of hydrogen protons, to make ATP. That's chemiosmosis. But the actual traveling of the electrons happens prior to that. That's where the electron transport chain is. So you have 2 processes going up here. Is that clear? All right. I saw this someplace. It's kind of, if you look at it, you'll see how the electron transport's using this mill kind of analogy. You can look at this and follow it, see where it goes. A photon goes up, or a photon light is over here, a photon light is over here, and it goes this way. So that's photosystem 1, photosystem 2. If it works for you, you could use that. Okay, just another analogy. So this is the same thing we talked about, but now you can go step by step. You follow like step 1, step 2, step 3. So again, this will be the stroma. This will be inside the thylakoids. Same thing we just did. It's busier though, but it's the same thing. This is where your photosystem 2 is. Here's your photosystem 1. There's an electron transport chain over here. There's a second electron transport chain. Light is going to come into photosystem photosystem 2. That's going to take water. An electron from the water will go into the system to go through here, but it will also split off and make sure that we have 2 hydrogen ions that stay here, and we also have oxygen molecules that will go out into the air. This is where the oxygen comes from that we need to breathe as humans. So every time water splits, another oxygen molecule goes out there, and more hydrogens get built in here. The electron is going to pass through here. As it passes through here, it makes these electron transport chains run. So it's going to force another hydrogen to be pumped into the thylakoid lumen, making the concentration inside the thylakoid much higher in hydrogen protons. You're going against the gradient. It's going to go from low concentration of hydrogen to a high concentration. That's active transport. The electrons will keep on going through. More light goes in here so that we can actually make the electron go that way. And with this second part over here, we can actually oxidize NADP to become NADPH. Meanwhile, the high levels of hydrogen ions over here can go out this ATP synthase. Every time that ATP synthase takes in a hydrogen proton, it clicks, it turns. And when it does, you make an ATP. Okay? So we are not using any ATPs with this whole thing. We're making ATPs, but we're not using it. We're using the solar power as energy to make ATPs and NADPHs. Now we made a pot load of ATP and a pot load of NADPHs. Now these things are stored energy. They will now go into our second part of photosynthesis, the Calvin cycle, that is going to be used to make glucose or any sugar. Questions so far? It's plants. I can't make it any more enthusiastic than